Hello, my dear students. Today the topic of our lecture is the water and mineral metabolism. We will discuss the role of the water and the role of the mineral components, mineral compounds. Okay, let's start. The water. In adults, water content is about 70 till 75% of the body weight. In newborn, approximately 80% more. And in aging, water content is decreased, and approximately 50% of the weight of the body is the water. It means the water is the main compound of our body. Okay, the water. Brain is approximately on 80% consists of water. Heat approximately 78, and other organs, the water content you could see on this slide. Saliva 95, skin 70%. Biological role of the water and the functions. At first, water is a universal solvent because the water molecule is a dipole, and it could solve the polar compounds, the salts, the organic polar compounds, inorganic polar compounds. Okay, the second function is contributes to dissociation of the substances, especially weak electrolytes. Dissociation means the producers of the ions from the neutral charged molecule. Okay, next function is participates in biopolymer native structure formation and stabilization. It means, for example, the formation of the tertiary and structure of the proteins. Again, participates in metabolism reactions. It used for hydrolysis and hydration. Hydrolysis means when we break down the chemical bond with the aid of the water, and hydration means the addition of the water molecule to the compound. Next function, structural function. It means a part of the supramolecular structures. And also mechanical function, aids to the cells and intercellular substance turgor. It means formation of turgors of the connective tissue. And also transport function. Transport, as you could understand, it means provide the transport from one organ to another organ. And also heat balance regulator. Water requirement. For adults, approximately from 30 to 40 grams per kilogram of the body weight every day. And for children, it's more, 3 or 4 times more. And this slide illustrates the average intake per day. Whole amount is 2.5 liters, and among them 1.5 liters we could receive from different drinks, beverages, approximately 60%. The water intake with the food, approximately 30%, and by metabolism intake is approximately 10%, and this 10% approximately 250 milliliters, is provided by the complete aerobic oxidation of the fuel molecules, the so-called metabolic water. For example, fast 1 gram of fats could provide, after full oxidation, 1.5 milliliters of water. Full oxidation of carbohydrates, 1 gram of carbohydrates, could provide 0.6 milliliters of water, and full oxidation of 1 gram of proteins provide approximately 0.5 milliliters of water, and this amount is approximately 250 milliliters, the metabolic water. And average output per day, approximately 1.5 liters. We excreted with urine, also 700 milliliters accessible loss via skin and lungs, and with sweat the output per day approximately 200 milliliters, and with feces approximately 100 milliliters. And without water intake, human vital functions are maintained for 7 or 10 days, and loss of 20% of water in the body is fatal, and it means the water is very important for our body, and every day we should eat, we should drink the water. Okay, this slide illustrates the water distribution in the body, and you could see that approximately 2/3 of this water is the intracellular fluid water, and 1/3 is the extracellular fluid water. And among this amount, approximately 20% is the extravasal fluid, and intravasal fluid is approximately 5%. And among them, it could be the blood vessel fluid and also lymphatic fluid, approximately 1 to 10%. And the determining factor in the distribution of the water is the extra and intracellular space, is the osmolarity. This physical is the constant, and approximately 285 till 303 millimoles per liter. And osmolarity provides the distribution of the water. The exchange of water between intra and extravasal fluids is limited by oncotic and hydrodynamic blood pressure. And maybe you know that oncotic pressure is determined by the presence of proteins in the blood. The main regulator of water excretion from our body is the hormone vasopressin, and the main regulator of water intake in the body is the feeling of thirst. And now some words about the hormone vasopressin. Vasopressin, another name, is the antidiuretic hormone, the neurohypothalamus hormone. This hormone is the peptide hormone and consists of 9 amino acids. And in this case, we tell nanopeptide, and synthesis in the hypothalamus, supraoptic and paraventricular nucleus. Okay, and some words about the regulation of vasopressin secretion. It could be two causes. by the increasing of the osmotic blood pressure by 1% or more, and the second cause is the decreasing of the blood volume more than 10%. And increasing of the osmotic blood pressure and osmoreceptors reacted on this increasing, and the result is the secreting of the vasopressin. And the decreasing of the blood volume is determined by the volume receptors or baroreceptors on our cells, and again vasopressin secretion. And vasopressin acts on the kidney collecting ducts and stimulate the reabsorption of the water. And how vasopressin provides the reabsorption of the water? Okay, this figure illustrates the kidney collecting ducts. Okay. Here we could see the vasopressin receptor, 7 TMS receptor, V2 vasopressin receptor. Okay. Vasopressin binds with its receptor on the collecting ducts of the nephron. Then it causes the activation of adenylate cyclase, and this enzyme provides the transformation of ATP molecule to cAMP, the secondary messenger. And then this compound provides the activation of the protein kinase A. Protein kinase A, this is the enzyme that causes the phosphorylation, and this phosphorylation stimulates the movement and incorporation of a special transporters. The name of these special transporters is the aquaporins. AQP means the aquaporin. These transporters incorporated into the luminal membrane of the cells of the collecting ducts and stimulates the water excretion, the reabsorption of the water. And you could see. And with mutation of the gene encoding of the synthesis of aquaporin, second aquaporin, this number means the type of the aquaporin 2, provides the nephrogenic diabetes insipidus. Some words about aquaporins. This is the integral membrane proteins, and integral means that this protein penetrate through the whole membrane, and this protein is the homotetramers. Tetramers means that there are 4 subunits in this protein, and homo means that these subunits are equals each others. Each monomer forms a water channel, and each monomer penetrates the membrane 6 times. You could see on this page, on this figure. This is a cell membrane, and this is the monomer of the aquaporin. And you could see that this protein penetrates the membrane 1, 2, 3, 4, 5, 6 times. And conservative sequence from asparagine, proline, and alanine are involved in this formation of this channel. Here you could see this specific sequence of amino acids, and each monomer provides the water channel, and this aquaporine is the combination of 4 equals proteins, and the result is the formation of the homotetramer, and this is protein is the aquaporine, and this aquaporine provides the penetration of water from extracellular to the cytoplasm. This is the channel for water. And there are some types of these proteins. The numbering starts from 0 till 20, found in humans. And there are 2 subfamilies. The first of them is only water, is allowed through, is just for water. And the second subfamily is provides a penetration for the water and other small uncharged molecules, for example, glycerin or urea, and some clinical aspects about aquaporins. The defect of the type 2 of aquaporine provides the nephrogenic diabetes insipidus. The defect of aquaporine number 9 provides the pathological state glaucoma, and other pathological state you could see provided by the problems with the aquaporins. And this is the photo of the Peter Agree, and he discovered these proteins, aquaporins, and received the Nobel Prize in Chemistry some years ago. And some words about the diabetes insipidus. Causes. It could be caused by nephrogenic reasons and central problem. Nephrogenic could be caused by the decreasing amount or defect of vasopressin receptors. And it's known that more than 100 vasopressin with 2 receptor mutations. And the second reason could be the decreased amounts or defect of renal aquaporins. And the central causes, the damage of the pituitary gland and hypothalamus. And this pathological state is usually the main features are the polyuria, more than 10 liters per 1 day, more than 10 liters per 1 day. The usual amount approximately 2 liters. But during this pathological state, we have now the reabsorption of the water, and this water secreted with the urine, and the whole amount of this water is more than 10 liters. The second feature is the hypostenuria. It's 1.001 to 1.004. It means the density of approximately pure water. The urine is like the pure water. And another is the polydipsia, or in other words is the thirst. Because we delete a lot amount of water, and we should drink it. Okay, and now about minerals. All minerals, depending on the concentration in the body, are divided into 2 classes. The macro elements at first, and the micro elements, or another name is trace elements. Macro elements, minerals, the major minerals, are the minerals the content of which exceeds 50 milligrams per kilogram, or more than 10⁻³ of the body weight. and among them there are seven major elements: sodium, potassium, there are the classical electrolytes, calcium, magnesium, phosphorus, sulfur, and chlorine. Chlorine is the microelement. And we start with the sodium. And the sodium is the major extracellular cation. The normal concentration of sodium in the blood plasma is in this range, 135 to 150 millimoles per liter. And the body contains approximately 100 grams of sodium. And this sodium is approximately 50% in extracellular fluid, 40% in bones and cartilages, and 10% in the cells. And excretion of the sodium. The 95% excreted with urine, and only 5% excreted with sweat and through the intestines. The human body can excrete approximately 7 grams per day of sodium chloride, and usually from 5 to 15 grams of sodium chloride per day comes to our organism with food. And norm of sodium chloride for hypertensive patients no more than 5 grams per day. The biological role of the sodium. At first, this mineral regulates the volume of the extracellular fluid and also regulates the osmotic pressure. The second one regulates the body's acid-base balance, because this cation could be exchanged with protons in the kidneys. And the protons is the acid, and it means these sodium cations could regulate the acid-base balance. Also participates in formation of electric membranous potential, sodium, potassium, and these two cations regulate this membranous potential. And also it is involved in the intestinal absorption of some molecules, for example glucose, galactose, and also amino acids, by the mechanism of same port. And daily requirements of this mineral. The main source of the sodium is the salt, sodium chloride, and you should eat not more than 5 to 8 grams of sodium chloride. And the main sources of sodium is the salt, also the salty fish, and other products. And there are some states, 2 states, when we have a lot of sodium in our body, in the blood, or less than normal. If less, in this state, the sodium deficiency provides a hyponatremia. Hypo means less than normal. And it could be caused by the reimbursement only with water for fluid losses during vomiting and diarrhea. Also, it could be caused by the use of diuretics or fasting. Also salt-free diet provides a lot less than normal amount of the sodium in our body. Also, kidney disease with impaired reabsorption and sometimes, for example, Addison's disease. And some words about this pathological state, about this Addison's disease. And this Addison's disease, or another name is the Bronze disease or adrenal insufficiency, could be caused by next problems. It could be caused by tuberculosis or autoimmune damages. Also, the decreasing amount of cortisol and aldosterone, and increasing level of adrenocorticotropic hormone. And there are some symptoms this figure illustrate of this disease. And the main of them is the bronze pigmentation of skin. You could see that the usual color is white, and the pigmentation like bronze color is the characteristic for this disease. And again, this figure illustrates the pigmentation of the. Also, it could be observed changes in distribution of body hair, for example, for women. Also, some problems with gastric tract disturbance, also weakness, hypoglycemia, and hypertension and weight loss. Symptoms, as I said, loss of appetite, hypotension, nausea, tachycardia, muscle weakness, and mental disorders by the hyponatremia. And the opposite state, the excess of sodium. The name of this state is the hypernatremia. And it could be caused by heavy sweetening, sufficient drinking with an excess of salty food, also provided by diabetes insipidus, and Crohn's disease or hyperaldosteronism. Hyperaldosteronism, we will set then, okay, and also Cushing's syndrome. And symptoms of this pathological state, hypernatremia, are hypertension, edema, depression, anxiety, and fear. Okay, next mineral is the potassium. Potassium is the main intracellular cation. The sodium is the extracellular cation. Potassium is the intracellular cation. The normal concentration of sodium is 3.4 till 5.6 millimole per liter. It's easy for remembering 3, 4, 5, 6. The numbers. The body contains 150 till 170 grams of sodium, and the approximately 98% of them is intracellular, and approximately 2% is extracellularly. And about the biological role of the potassium. At first, this cation takes part in the formation and maintenance of osmotic pressure in cells, because this cation is inside the cells. The second one maintains again acid-base balance as part of intracellular buffer system. For example, hemoglobin, oxygenated hemoglobin, compared to sodium, which is part of the blood buffer systems. Again, intracellular And also the side function it could be precipitate information of electric membranous potential with sodium. Then regulator of neuromuscular conductivity, contractility. The day requirement. The day requirement you could see here approximately 3 grams per day, less than sodium, and the main daily sources of this product you could see on this figure, or tomatoes and other products. And again, there are two states when we have a lot amount of potassium and less than normal. If less than normal, it will be hypokalemia, arrhythmias and slowing of intracardiac conduction, cardiac arrest in the diastole phase, the so named silent death. Also, nausea, anorexia, muscle weakness, and intestinal atony. And the opposite state is the hyperkalemia. It could be caused by paresthesias, muscle paralysis. Also, some changes in cardiogram, for example, bradycardia. Heat block could be observed at sodium potassium concentration more than 10 millimoles per liter, and cardiac arrest if the potassium concentration more than 20 millimoles per liter. I remember you that I again say you that the normal concentration is approximately 4 or 5 millimole per liter, and if the potassium concentration increased in 2 times, it provides a heat block, and if the potassium concentration increased in 5 times, it provides a cardiac arrest. And again, this slide illustrates the comparison between sodium and potassium. You could see that in extracellular fluid the main cation is the sodium, and intracellular fluid the main cation is the potassium. And the difference in the concentration of monovalent cations, sodium and potassium, on cell membrane is maintained by a special enzyme, a special pump, sodium potassium ATPase. This concentration is maintained by this enzyme. And some words about this pump or this enzyme, sodium potassium ATPase. This enzyme was opened in 1957. This is the integrated protein, the glycoprotein. This figure illustrates the structure. It consists of 2 subunits, the alpha subunit and beta subunit. This is the proteins, and these lines is the carbohydrates, and the complex of them is the glycoprotein. The combination of the protein and carbohydrates provides the glycoprotein. And this is the name, here you could see the name of the scientist that discovered this pump, this enzyme, James Christian School. And in 1997 he received the Nobel Prize in Chemistry. And this slide illustrates how this enzyme is working. Okay, this is the cell membrane. Here is the extracellular fluid, this is the cytoplasm. Okay, in the cytoplasm the sodium concentration is low and the potassium is high. But in the extracellular fluid, the opposite situation. The concentration of the sodium is high, the potassium concentration is low. And the task of this enzyme is the pumping of sodium from the cytoplasm to intracellular fluid and the potassium from extracellular to the cytoplasm. You could see that there are some states of this enzyme. The open state in the cytoplasm, and this state provides the addition of 3 sodium cations on the specific sites of this enzyme. Then we have the phosphorylation of this enzyme. You could see that this enzyme is without phosphoric acid residue, but next step is the phosphorylation. This phosphoric acid residues comes from ATP, and phosphorylation causes the closing for cytosol and opening of this enzyme into extracellular fluid. And during this opening, the sodium cations penetrate to the extracellular fluid. And next stage is the binding with 2 potassium ions. Then next stage is the deletion of this phosphate group, and it causes the closing for extracellular fluid and opening to the cytoplasm. And this opening provides the penetration of potassium ions to cytoplasm. And the cycle could be started again. We use the energy of ATP for pumping the sodium cations from the cytoplasm to extracellular fluids with low sodium concentration in the area with the high sodium concentration. And the opposite process. The potassium ions from the area with low sodium concentration will be pumping to the cytoplasm, where the potassium concentration is high. And for this, we use the energy of ATP. And activators of this enzyme is the CAMP. And also it could be find it that we could inhibit the work of this enzyme. And among the inhibitors are the specific compounds wobbein and also cardiac glycosides. The name, for example, digoxin and strophanthin. These cardiac glycosides could be received from plant digitalis, for example, and these inhibitors, cardiac glycosides, inhibit the sodium potassium ATP phase. And for example, for heat cell, myocardial cell, we could see that inhibiting of this enzyme provides the increasing of the sodium concentration in the cell, because this pump doesn't work, and it means the sodium stay in the cell. And another pump, sodium calcium exchanger, is work as a symport. This pump work as the antiport. It means sodium excreted, the potassium penetrate to the cells, but this is their myth. And that provides the increasing of the calcium concentration in the myocardial cell, and this calcium binds with the troponin C. This is the protein of the contractility, and this binding provides the so-called inotropy effect. It means the cell is good, is work very good, and its cardiac glycosides used as drugs for the people with problems with heart. Okay, some slides. Okay, next mineral is the chlorine or chloride ion. Chloride ion is a major extracellular anion. The major extracellular cation is sodium, and the chloride is the major extracellular anion. This ion, the body content of the chloride is about 30 millimoles per kilogram of body weight, and a lot of the amount of the chloride ion, approximately 88%, are inside the cells and 12% outside the cells. The normal concentration of chloride in the blood plasma is from approximately 100 millimole per liter. The biological role of this microelement. At first, participates in the regulation of osmotic pressure and water balance. Also, this ion, chloride ion, is used for gastric juice formation. Also, as a component of sodium chloride, is essential for acid-base balance regulation. And also, this ion could activate some enzymes, for example, amylase activator. Okay, I understand that it was a mistake in the previous slide, and as I said, the chloride is the major extracellular anion, and it means that the main amount, approximately 88% of the total chloride, are outside, it's a mistake, outside the cells, and 12% is inside the cells. I'm so sorry, it's a mistake. It should be different. Okay, biological role we discussed, and now the products in which we could find the chlorides. The daily requirement is not more than 5 to 8 grams of sodium chloride, the usual salt. And this chloride ion is connected with sodium, and for sources you could see the products with the sodium. Bread and salt fish and so on. And there are big problems with chloride channels, could be. And the genetic defect of chloride channel, the name of this chloride channel is the CFTR, cystic fibrosis transmembrane conductance regulator, and results is the cystic fibrosis. And the viscosity of the secretions of the exocrine glands is increased. There is no water. and the viscosity is increased, and lesions of the lungs, liver, pancreas, and reproductive trunks also could be observed. And now about the regulation of electrolyte and water balance. Electrolytes and water balance are regulated together. It's combined, and the kidney play a predominant role in these regards. The regulation is mostly achieved through some hormones. For example, atrial natriuretic factor, vasopressin, or another name is antidiuretic hormone, and aldosterone. It's the mineral corticoid. The main three hormones: atrial natriuretic factor, vasopressin, and aldosterone. At first, atrial natriuretic factor is a polypeptide hormone, and this hormone is secreted by the right atrium of the heart in response to signals such as blood volume increase and high salt intake and increased right atrial pressure. And it means the name of this hormone is from this atrial pressure, atrial. Okay, then this hormone binds to its 1 TMS receptor, not 7, 1 TMS receptor, and its intracellular domain has a guanylate cyclase activity. This receptor consists only one peptide chain that penetrates the membrane, and the cytoplasmic domain, intracellular domain, has guanylate cyclase activity. And the result is after binding of the receptor with this hormone, is the activation of this enzyme, guanylate cyclase. And this enzyme, guanylate cyclase, convert GTP molecules to cGMP, cyclic guanylate monophosphate. And this secondary messenger, cGMP, activates enzyme protein kinase G, which then phosphorylates other cellular proteins involved in this pathway. And this hormone stimulates the glomerular filtration and increases excretion of sodium, and with sodium chloride ions, and also water. Okay, increased blood volume, increasing high salt intake, and increased right atrial and increased pressure. And after the hormone secretion, we have the excretion of the water, of the excess of sodium, and the excess of chloride ions. And this picture illustrates the action of this NP factor. Next hormone is the vasopressin. This is the antidiuretic hormone. And as I said earlier, these hormones, after binding with the special receptors in distal collecting tubes of nephron, activates through adenylate cyclase formation of cAMP. This secondary messenger activates protein kinase A, and then stimulation of aquaporin 2 translocation and its incorporation in the luminal membrane of the collecting tubes. And the result is the water reabsorption from distal kidney tubes increased significantly. Okay, this hormone vasopressin is only is regulate the water reabsorption, not mineral, just water. And next hormone, aldosterone, regulates the mineral reabsorption and secretion. And this hormone activate, the role of this hormone is the activation of the sodium-potassium ATP phase synthesis, the special enzyme, the special pump, mineral pump, and the result of this activation is the sodium and water reabsorption in approximately 12 to 24 hours period. Again, it stimulates the chloride and hydrocarbonate reabsorption in kidneys. And if this hormone reabsorbs sodium, the opposite action is the excretion of the potassium and protons excretion, reabsorption of sodium and potassium excretion. And also this hormone increases the arterial pressure, because this hormone stimulates the sodium absorption and water again. And this big scheme illustrates the so-called renin, angiotensin, aldosterone system, RAS system. And this system conjugate together the action of some organs in the regulation of the aldosterone secretion and regulation of water and mineral balance. Okay, we have two organs, liver and kidney. Liver secretes angiotensinogen, a protein, and kidney secrete another protein. Kidney secretes the protein, and the name of this protein is renin. And renin is the enzyme. And this enzyme could act on angiotensinogen and convert this angiotensinogen to angiotensin 1. Renin converts angiotensinogen to angiotensin 1. Then we have a lungs. This organ secrete angiotensin conversion enzyme, and this enzyme, angiotensin conversion enzyme, could convert angiotensin 1 to angiotensin 2. The main reaction is the proteolysis in this case. Okay, and angiotensin 2 binds with its receptors and causes, for example, angiotensin stimulates aldosterone secretion in the adrenal glands. And aldosterone promotes sodium and fluid retention, reabsorption. Sodium reabsorption, as I said earlier, potassium and protons excretion. Next action, angiotensin stimulates sodium and fluid retention in the kidneys, as I said. Also, angiotensin stimulates muscle hypertrophy and fibrosis in the heart. Also, angiotensin stimulates sympathetic overflow in the brain, and also angiotensin stimulates vasoconstriction in blood vessels. And the result is the increasing of the arterial pressure, the blood pressure. And also you could see the names of some inhibitors of these enzymes. For example, aliskiren or tecturna blocks renin. blocks renin, and it means this renin couldn't convert angiotensinogen to angiotensin 1, and it could be used for hypertensive patients. And another inhibitors of this enzyme, angiotensin conversion enzyme, ASE inhibitors, block this angiotensin conversion enzyme, and the result is that angiotensin 2 couldn't be synthesized. And the result, again, there is no the secretion of the aldosterone, there is no the reabsorption of the sodium and water, and there is no the increasing of the blood pressure. And if you could see, the signal for renin secretion by the kidneys could be, for example, the decreased arterial pressure in the kidneys at first. It could be the decreased sodium in the blood, and it also could be increased sympathetic tone. And also from this figure you could see that angiotensin 2 receptors are located in adrenal glands, vascular smooth muscles, the heart, and in the brain. And the conjugated action of this system, the name of the system is renin-angiotensin-aldosterone system, provides the water-mineral balance in our body. Okay, next lecture will be about the calcium and magnesium and microelements. And okay, thank you for your attention and good luck.